really is absolutely fantastic to be here presenting, because I think this initiative is fantastic and it really gets to such a big audience as well. So big thanks again to the organisers for allowing me to come and tell you a little bit about what we're up to in the team, and that's mostly exploring early life microbiome dynamics. So let's get started, just to make sure that we've got plenty of time for questions at the end. So I will start with the acknowledgements, because I have a tendency to keep chatting, which may mean I don't get to the end of the presentation. I just want to say that really all of the work I am presenting is underpinned by the fantastic teams that we've had across the years, and also our fantastic collaborators as well. Many of them are healthcare professionals and clinicians, and really we could not do this work without them. So I'm going to take you on a little journey, kind of, I guess, exploring some of the stories that we've been doing across the years, and also some very recent work that's been pre-printed and actually has very recently been accepted. And of course, very happy to answer any questions, including any follow-up questions as well. And obviously, thank you very much to our funders as well, because without them, we can't do the cool science that we do. So we've got a really big interest in the team in understanding how the early life microbiome kind of overlaps with healthy host development. Because of course, during the first 1,000 days, and that's really from conception up to 2 years of age, this is when obviously many different host features are kind of getting up and running. So like immune, metabolic, and neural, for example. But of course, it overlaps also when our microbial ecosystems are first seeded and then establish and mature over those first 1,000 days. And of course, what happens in pregnancy and birth and shortly after birth doesn't just kind of set things up kind of short term. It really has massive impacts longer term for overall health and well-being as well. So we're really interested in the team and understanding what happens to the microbiome during this key developmental window, and really how can we harness this knowledge to think about maybe translational or intervention strategies for particularly those, I guess, individuals most at risk. So we've got a really big focus in the team on lots of different microorganisms, but I would say we're mostly bacteriologists, although we do a little bit of virome work and phage, and also delving into the microbiome. But we've got a really big focus on bifidobacterium, and we love BIF for a number of different reasons. Kind of a microbial superstar, but we're really interested in BIF because it's one of the first colonizers after birth, and it isn't just found in babies. We hopefully have it persisting with us the whole way through the life course as well. But with an early life focus, I think what's really important to highlight here is that they really are seeded at birth, and they're found at very high levels across this first 1,000 days that I mentioned. And actually, because they're found at really high levels across this developmental and maturation period, they're associated with health. Of course, defining what health is one of the huge challenges with microbiome research in general. But we know that BIF is also really important for actually immune priming. I'm not going to talk very much about that here. That's kind of other work, and I'm conscious about the time that I have, but also happy to answer any questions more generally if anybody has them. The one amazing cool thing about BIF and why it's found at really high levels during early life, and we will touch upon this a little bit, is it's exquisitely adapted to the early life diet. This is why if you take a poo sample from a breastfed vaginally delivered babies, it will be chock-a-block full of bifidobacterium, because they're able to metabolize some of the milk sugars that we have in human milk. Whereas formula-fed babies, we might have much lower levels of bifidobacterium. And that obviously has consequences in terms of that immune maturation and maybe risk for example, atopic diseases, both short and longer term. From a really exciting translational perspective, many species have actually been used for many years, actually as nutritional supplements, because they have the generally recognized as safe designation, which means that you can go in and basically start supplementing them. Yes, I'm absolutely conscious that there's other kind of things that have happened kind of in the last kind of 18 months. And again, we can talk about that a little bit more in the Q&A if that's of relevance, but really they have an excellent safety track record. The BIF is amazing, but on the flip side of that, it's incredibly susceptible to some of the external factors that might perturb these burgeoning early life microbial communities. And I'll talk a little bit about that on the next slide. So they're really susceptible to, for example, if you have a course of antibiotics, because they're often or very rarely antibiotic resistant. And because they have adapted to specific dietary components, if we change that diet, we might change the abundances of bifidobacterium. And actually low levels of BIF have been implicated in a whole host of different kind of disease and conditions. But again, for this talk, we're going to stick with kind of the neonatal side of things. So we do love BIF, but who else hangs out with bifidobacterium? And I'm just introducing this because I'm going to talk about this at the very end of the presentation. And one other thing I just want to highlight here, although given the audience, I think probably you're all well aware. I think one thing just from a general communication perspective, when we talk about the microbiome, is that we use terms like high diversity being a good thing. Now, that's not always true. In early life, we actually want low overall microbial diversity because we want lots of bifidobacterium present. That's particularly in the gut. And I guess another example of that would be the vaginal microbiome, where we actually don't want a crazy diverse microbiome. We just want a lactobacillus dominated one. So that's just important just to highlight those differences there, because obviously that plays into the whole ecosystem structuring side of things. So other key taxa that we and many others have seen that also are kind of co-abundant with bifidobacterium during our life, particularly during kind of the milk phases of feeding, would be Bacteroides, Streptococcus, Blautia, and Escherichia coli or E. coli. And E. coli is an interesting one because many people would think of that as a bona fide pathogen and potentially always being associated with disease. But actually in some contexts, E. coli is actually really important. And that's kind of, I'm going to come back to that, but I just wanted to introduce that topic here. So we have our host development, we have our microbiome, but like all ecosystems, we want these to kind of establish in a particular trajectory, and we need to be really careful about not disturbing or perturbing them. And early life is a tricky time because there's a whole host of different external factors that might shift those initial microbial communities, including our keystone bifidobacterium. So depending on how you're born, so for example, and many people on this call, including some of our work, have indicated that, cesarean section delivered babies have a different microbiota in comparison to vaginally delivered babies. Babies get shed loads of antibiotics. I won't stress that point because we'll get onto that, but this may also have an influence on these early life microbial communities, gestational age, and also I've already mentioned diet, because of course diet we know has a massive impact on what happens to the microbial communities particularly within the gut. And I think the important thing just to highlight here is that all of these different factors could happen by themselves or together. But in terms of a group of early life, I guess, participants or patients would be premature babies. And multiple factors are actually found from a perturbation perspective in these premature babies. And I'm just going to give you a couple of examples of some of the studies that we've been doing over the years on these very fragile at-risk populations. So premature babies are those defined as less than 37 weeks gestation. They're often very low birth weight, so less than 1.5 kilograms. Full-term babies might be 3.5, 4, but of course there's always a range. And actually some of our premature babies that we've had on our studies is as tiny as 500 grams, so like a bag of sugar. Preterm birth rates vary widely, kind of across countries and actually even within country as well. But I think the important thing to highlight here is that preterm birth complications are the leading cause of death among children under 5. And actually our kind of, as a group of individuals and kind of more globally, actually chipping away at those numbers has actually proved to be really difficult. And it's something which is why we need to focus on this so carefully. There's many different infections that cause a problem in premature baby in terms of morbidity and mortality. Just a couple of these are listed here. There's normally not one kind of microorganism that might be associated with a particular condition. There's normally lots of different ones, but I'm going to give some specific examples just as we go through the presentation. But they're associated with a whole range of different infection readouts, as you can see here. But going back to these factors that perturb microbial communities, cesarean section is really common in premature infant cohorts. They reside in sterile neonatal intensive care units. They often stay in these intensive care units for very long periods of time, 6 weeks or even up to a year, depending on how early they're born. and they all get a shed load of antibiotics, but we'll come back to that. And this basically means premature babies have a radically different microbial composition in comparison to their full-term counterparts. So they're often kind of colonized with microorganisms that have pathogenic potential, or so-called pathobionts, and they're often missing keystone, healthy, immune and colonization resistance promoting bifidobacterium. And this is really important because one of the main roles our microbiome does for us is colonization resistance. And because the preterm infant doesn't have those same microbial communities and those similar species and strains, this means they have very reduced colonization resistance. And this is important for a whole host of different things, but just in terms of time for the presentation, it's really important for a very devastating gastrointestinal emergency that you get in premature babies called NEC, or necrotizing enterocolitis. About 5 to 15% of premature babies can have this, but this again varies widely, kind of globally and within country as well. Up to 40% kind of in terms of death rates, mortality, and there's really serious long-term health implications for those babies that do get NEC, but actually come out the other end as well. And importantly, NEC is multifactorial. There's lots of other things going on, but it is also linked to overgrowth of microbes that might be residing in the premature baby gut. This is just some of the clinical presentations, and I'll show some of these slides or images a little bit later. But the one thing I want to highlight here is that this is a very rapid onset disease. So the babies will present very, very quickly, maybe as quick between kind of 4 to 8 hours, and present with a distended abdomen. If you do an X-ray, you can see kind of gas cysts within the intestine called pneumocystis intestinalis, and then you physically get bowel necrosis. And this is where your neonatal surgeon comes in, hopefully gets rid of the necrotic parts of the bowel, sews up the healthy parts of the bowel, but you've just taken a whopping big part of the premature baby colon out, which is why we have these longer term health implications as well. And importantly, if you kind of chat to kind of nurses and clinicians in neonatal intensive care units, they have outbreaks of NEC, which would suggest an infectious kind of agent is involved. And there's a number of different pathogens associated with NEC, but I'm just going to tell you a little bit about some of the stories that we've been working up with respect to Clostridium perfringens. And actually, if you go back and look at the medical literature, even kind of back to the 50s, you can actually see that preterm NEC, in terms of some of the textbooks, is actually called gas gangrene of the bowel. And that's really important because Clostridium perfringens is the agent for tissue necrosis or gas gangrene. So kind of learning, I guess, some of the different facets from what's been done previously and putting that, I guess, in terms of thinking about new patient population. So we know that perfringens has been linked to NEC, and we've known that for a number of years. And in fact, it's established in terms of pathogenesis and lots of other neonatal animal gut diseases. So that's why important. One, health is really important, making sure that we don't work in our silos in terms of human microbiome, but thinking more generally about how microorganisms are really important for basically everything. But it's only really since we've started kind of collecting baby poo samples longitudinally from premature babies that we've really got to handle on the fact that there might be this overgrowth of pathogenic microbes. And some really elegant work done with Simon Kroll's team a number of years ago kind of showed that there was other bacteria involved in NEC, actually including Klebsiella. But another cohort of those babies had this bloom in this kind of red, which you can see here, which is Clostridium and Clostridium perfringens. And this is Raymond. Raymond's a fantastic postdoc in the team. And Raymond's been working really, really focused on Clostridium perfringens for a number of years. And I just want to highlight some of his work over the next couple of slides. So instead of actually doing microbiome for the next kind of bit that I'm going to share with you, I think the other thing that's really important is that we can sequence everything and have a look what's there, and that's fab and it's exciting, but it's also really exciting to do the culturing, get our hands on these microorganisms so we can actually really in-depth profile them, and also we can actually do some experiments with them as well. So looking at premature babies, and this was kind of a 5-year study over 5 neonatal intensive care units across the United Kingdom. Interestingly, if you take a preterm baby poo sample and you basically just go fishing for Clostridium perfringens, a quarter of those babies will have perfringens, but a quarter of those babies will not necessarily get NEC. So that's why Raymond was interested, because why is that the case? So taking our Clostridium perfringens isolates that have been taken from the preterm baby poo, doing whole genome sequencing, and then basically lining these up to kind of look at their relatedness, and then delving into some of the other genomic features that would be important in the context of NEC, including toxin production. Can they colonize? Are they antibiotic resistant? For example, a couple of interesting features came out from this. I'm not going to labor the point too much because this paper was published a couple of years ago, but again, I'm happy to answer any questions that anybody has. But I think one interesting thing came out of this particular study, and the fact that Clostridium perfringens is a known toxin producer, and in fact, it can produce up to 20 different toxins, although thankfully not one strain encodes all 20 toxins, because that would be terrifying. But there's one particular toxin here that we were seeing consistently in the C. perfringens isolates from the NEC babies that we were not seeing in the C. perfringens strains that we were finding from the premature babies that did not have NEC. And this gene is PFOA, and it encodes for the toxin perfringilysin O, which is a pore-forming toxin. And actually, presence of this toxin gene in the genome of perfringens, when you actually do some experiments with it, it linked back to a number of the different features that we see in premature babies. So for example, we saw increased necrosis when we looked at kind of epithelial monolayers in vitro, and it was the presence of the toxin that directly correlated with that. And then also doing kind of more preclinical models with mice, what we were able to show that actually the strains that encoded the toxin, there was kind of lots of kind of histopathology that we would also quite often see in babies that have NEC. And also we see a humongous immune infiltration as well, which was also associated with kind of a higher pathological score with respect to kind of gut slices that we were able to take from the intestines of the mice as well. So it's one of those ones where just having that one toxin gene versus not having it, and basically many other kind of genomic features being identical, was enough to have that very particular, I guess, virulence signature. And the other thing that I mentioned in terms of linking to these outbreaks of NEC, of course, one of the things that you can do when you have whole genome sequences is you can do genomic epidemiology, which is super fun and super cool. And hopefully what you can see here is you can see a straight line here on the phylogenetic tree that I've just brought over from the other slide. And actually Raymond, doing a bit of detective work, realized that this was over 30 strains of perfringens that were basically clonal. And what I mean by that is there was one SNP or single nucleotide polymorphism different between those 31 strains, but those 31 strains were distributed across 6 babies and across 2 hospitals and across a 100-day window. So that suggests that they're moving around. And in fact, Raymond was able to show that, so able to show kind of the first almost index case of perfringens that actually caused, that was found in a set of twins. Luckily, they were treated with antibiotics very, very quickly. But unfortunately, one of the other premature babies on the ward got that strain, and they actually succumbed for NEC later. And then that same perfringen strains was then spread in a sister hospital when babies were moved. So just important from an infection prevention perspective. And the reason that we potentially see these outbreaks is Clostridium is one of these bacterial genera that can produce these environmentally resistant spores, which you can see here on the electron micrograph. And actually the strains from the babies that had NEC, those particular strains, as well as having the toxin, were actually better able to produce these spores, which would be really important from thinking about spreading around wards and then more generally as well. But a really super interesting study that came out really recently from Professor Chris Stewart's lab at Newcastle University is why it's so important to look at the strain level. And Chris and his team were able to actually show that Clostridium perfringens, in the context, if it doesn't have a toxin, might actually encode beneficial traits that's actually really important from an infection prevention perspective. And I just wanted to highlight that. That's kind of hot off the press. Please do go and have a read. It's absolutely fantastic paper. But it kind of just shows you the fact that just by having one simple difference in the genomes, either means it's going to potentially is going to kill you, or it's actually going to keep you healthy. So it's just important just to highlight that. So another thing that we've been really interested in is then thinking about premature babies have this reduced colonization resistance. They're at risk of overgrowth of pathogenic bacteria. So what can we do about that? So importantly here, we were really keen to see if we could just re-establish a healthy microbiota that looks like a full-term baby in premature babies and establish this strong infection or colonization resistance. So can we basically come in with our microbial pioneer, this beautiful Y-shaped bacterium here on the micrograph, a Bifidobacterium, so pretty. And that's kind of where our Bambi study came in. And again, I'm not going to talk about too much about this because this has been published, but I wanted to just introduce you because we've more recently followed up on this, and that's the next couple of slides I want to show you. So this was Christina and Matthew and the team that really led on this. And again, this was kind of a multi-clinical study, observational study, where we had premature babies that were routinely given a probiotic that included Lactobacillus acidophilus and Bifidobacterium bifidum, and they were kind of dosed twice daily. And then we took lots and lots of premature poo samples, and we basically wanted to look at babies that were not given a routine probiotic supplement. What did the microbiome look like, and why was that important from a clinical setting? So just in terms of top level from this, and actually because it was a while ago, and of course we started the study many years before that, this was obviously just using metataxonomic sequencing. The next couple of studies that I'll show you are on shotgun metagenomics. But what we were able to show here is that by giving the Bifidobacterium supplement, which is super cheap and which is important in the UK, we have free healthcare. So the National Health Service, the NHS, so it's really important to be cost effective. We see Bifidobacterium dominating our supplemented babies, and we see a concurrent decline in potentially microbes that might be associated with neck and or sepsis or other infection outcomes. And of course, from a clinical perspective, it's great to change the microbial ecosystem, but who cares to some extent? And that's what our clinical collaborators kind of asked. And working with Professor Paul Clark, who we've worked with for many years, fantastic consultant neonatologist, and Claire, who was a trainee clinician in the lab. And we were able to show by just giving this probiotic supplement in the unit routinely, we were able to reduce the incidence of necrotizing enterocolitis by over 50%, which was fantastic. And there's lots of work that's been followed up on that. So just in terms of kind of, I guess, the reboot of Bambi or Bambi 2.0, and Raymond kind of re-enters the stage here, is we were really interested to look to see if giving a probiotic might also influence, I guess, the taxonomic composition of the microbiome. But what does that do in the context of the resistome? And of course, antimicrobial resistance is a huge problem. So it's really important to understand what that might be happening in premature babies, which are incredibly susceptible to infection, as I introduced earlier, and also multi-drug resistant pathogens, okay? And this is important because premature babies in the UK are prophylactically given benzopenicillin or gentamicin at birth. So that's just kind of what happens in routine clinical care. But because we had so many premature baby poo samples, we were able to kind of start to select those poo samples and to sequence them so that we could specifically look at premature babies that had got, for example, probiotics or non-probiotics, and also those babies that had antibiotics and those that had not had antibiotics. And for all of us in the audience, we're very aware of all the confounding factors that sometimes make microbiome kind of analysis, drilling down to the mechanistic detail, really difficult. So we had a really nice opportunity to do this. And this was published just last year, but I'll give you some of the top highlights as well. So just like our Bambi study, we were able to show that Bifidobacterium really dominated the babies that were supplemented with the probiotic. And again, we showed that the babies that were dominated by Bifidobacterium also had a lower overall microbial diversity, which would correlate to what we see in rather full-term babies. And then if you kind of drill down to the species, you can see that the supplemented babies are actually colonized by Bifidobacterium bifidum, which is in the probiotic. And we also see that in the case of other Bifidobacterium, they're also encouraged into the ecosystem as well. Unsurprisingly, because I introduced the topic that Bifidobacterium is not often antimicrobial resistant, what we see is that if the preterm baby gets antibiotics, we reduce the Bifidobacterium load, which isn't fantastic, but that's why we give it on a regular basis. Interestingly, we actually show that the Bifidobacterium probiotic actually reduces the number of ARGs or antimicrobial resistance genes that we see in the premature baby gut, which is fantastic. And we can actually see that it's lots of different classes of antimicrobial resistance genes. that are reduced in the babies that get the supplement in comparison to the control babies that don't. And this includes some kind of last line antibiotics that might be given to premature babies if they're super sick and none of the other antibiotics are working. So we saw these antimicrobial resistance determinants like colistin, for example, and then non-supplemented babies, but we did not see them in the babies that received the standard probiotic. So in terms of who's carrying these antimicrobial resistance genes, I think that's really important to delve into. So Staphylococcus, Klebsiella, Enterococcus, and Escherichia carry most of these ARGs, which you can just see here. And this is just kind of a zoom in on the tree that Raymond did as part of this study. But I think what's really important to highlight here is many antimicrobial resistance genes are what we really need to worry about in the context of antimicrobial resistance is the fact that strains can swap stuff around. So horizontal gene transfer. And actually we found that antibiotics actually increase the rate of horizontal gene transfer. And we actually found that many of the antimicrobial resistance genes were encoded in plasmids. And actually we found more plasmids associated with multi-drug resistant bacteria in the babies that had not had the probiotic supplement. So that's just really important. And I'll kind of leave that story there because again, I'm a little bit conscious about time. But again, happy to answer any questions, and you can obviously read the paper to your heart's content and all the supplementary files. But one thing I just wanted to highlight here, because it's literally hot off the press and kind of brings us back to term babies. And again, thinking about nuances within the microbiome, but specifically in early life, is E. coli. So E. coli is an absolutely deadly pathogen and actually causes a whole host of different neonatal disease and infection outcomes. But as part of the study, interestingly, what we found was that actually in the babies that had got the probiotic supplement, E. coli was still there, but it just didn't carry these antimicrobial resistance genes. Whereas the babies that were really low in bifidobacterium, there was E. coli, but a lot of that E. coli was multi-drug resistant and also had lots of virulence factors that might be associated with infection. So what about this commensal E. coli? And I did say we would kind of get back to that. So this was a lovely collaboration with John and Niels from Maastricht University. And we were lucky enough, and that wasn't actually deliberate, we were lucky enough to actually work with some of the samples and their data from their Dutch lucky longitudinal cohort. And these to go back to full-term babies, healthy, vaginally delivered, breastfed infants. And this has been pre-printed and it's very recently been accepted in Nature Comms. And many people have already shown this, but what we have is we have obviously low diversity in early life, particularly when the babies are on a milk-based diet. And I'm going to talk about that just over the next 2 slides before I finish. But you can quite nicely see in the PCA plot here, these are the different ages. You can see that there's more dissimilar at these earlier time points. There's this convergence kind of around kind of moving up to 11 months post-birth. And this is mum here. So you can kind of see this trajectory here. And this very much links with diet. And many other people have shown that. But something just to kind of highlight here, which is maybe of interest to people on the call, is who's in these ecosystems and who's hanging out. So interesting, this is David, a fantastic postdoc in the lab when we were in TU München in Germany. David was able to show that actually when BIF is dominant, we get E. coli. And we can see this very, very clearly in this first kind of 6 months. And this overlaps with the milk-based diet. And in fact, all of the babies on the lucky cohort for these ones had all received maternal milk rather than formula. And then we see when we're not having as much bifidobacterium, we don't necessarily kind of see this hanging out together as well. And that's really interesting because as long as human milk is supplied, so across this first 2 to 6 months, E. coli is consistently prevalent with BIF. And the BIF that's consistently prevalent with would be Bifidobacterium bifidum, which I'll talk about just in a moment because that's quite a cool BIF. I mean, they're all cool, but bifidum's even more cool. Bifidobacterium longum, subspecies longum, and Bifidobacterium brevi. So the question is, why, if BIF is there, why do we see E. coli? Why do we see that kind of co-occurrence? And actually, this is really important because we see this overlap during that milk window. And actually, lactose, which is the simple sugar that we find in milk, and also human milk oligosaccharides, which are these slightly more complex sugars that we get in human milk, really are the primary carbon sources that are available for these microbial pioneers to start kind of degrading. And actually a really nice way that David was able to work with this, including Shaul, who was a fantastic collaborator as well on this, was to think about, rather than kind of annotating the metagenome assembled genomes that were pulled out of the shotgun metagenomic data, can we do kind of a annotation agnostic way to see if we have an either non-random or random kind of metabolic strategies that might indicate why E. coli and bifidobacterium are hanging out together. And what's important to highlight here, and this is just kind of the graph here, is that it looks like there's really distinct ecological strategies emerging between E. coli and bifidobacterium bifidum. So if it was kind of non-random, everything would follow on this diagonal line, but we see this kind of non-random spatial kind of separation here. And this suggests that actually there's maybe a little bit of functional association here. And I'm just about ready to finish off. But why do we see this? So E. coli will scavenge any residual lactose that makes down to, for example, the large intestine, because many of that is absorbed in the upper small intestine. But obviously, human milk oligosaccharides are also degraded predominantly by bifidobacterium. But actually, there's some bifidobacterium, including bifidobacterium bifidum, that all of the metabolic machinery to break down HMOs are actually on the surface of the bacterial cell. So this would suggest that if your enzyme cuts one of the side chains off these more complex sugar side chains, does that mean that those simple sugars are then available for E. coli to potentially hoover up? But the question is, what does bifidobacterium get out of it? Because does E. coli just kind of, because we keep seeing this co-association, so you would assume that they both get something out of this partnership. And it's kind of fairly big hint here about why this is. So David did some beautiful kind of mechanistic in vitro studies, basically showing that E. coli produces cysteine, but if you knock out the ability for E. coli to produce cysteine, bifidobacterium cannot grow. And on the flip side of that, the simple sugars that bifidobacterium basically produce after it starts snipping off and cutting down these more complex human milk oligosaccharides, the E. coli can potentially use these more simple sugars to grow. So you actually see this kind of really nice kind of cross-feeding mutualism, and it's maybe one of the reasons about why you see BIF and E. coli hanging out. And actually, if you start to look at infant data sets, it's super, super common. So actually, in this context, E. coli is not always bad, but actually it's really important for stabilizing bifidobacterium. And in fact, other people have done really elegant work showing E. coli is really important from an immune maturation perspective. And it just shows you why you've got to look at all these different aspects in detail. And I will stop there, and I'm really happy to answer any questions that anybody has. Thank you very much.